right.
I have nowhere to lose, so
There we go. Let's put the sound down. Great. And it is 9:30. Spot on. So welcome to a Neuroccino on this Monday morning, 9:30. We have a lot of new faces, so I'm looking forward to getting to know you a bit better. And today, Eduardo volunteered to present. Thank you very much for that. And we already have the link to the paper in the chat. So, Eduardo, if you're ready, take it away.
Thank you. Yeah, volunteering. I'm also on time.
Yeah, it's becoming a habit. I love it.
Okay, enough with the jokes. Can you see my screen?
Yes, if you zoom in a little bit, we can see it full screen. Yeah, perfect. Thank you.
Yeah, okay. Yeah, thanks, Stephanie. So, I have a very fresh paper for you today. It was just published like 15 days ago. And the main topic is yeah, like axon diameter and myelin thickness. So, of course, as we all know, the diameter of an axon and its myelinization is a key factor in determining, like, among many things, the speed of conduction of the neural signals. And larger and more myelinated axons are faster and also, on the downside, more expensive, and they require more space to be fitted in the brain. And a long-standing hypothesis slash assumption was that longer fibers tend to have larger calibers and are more myelinated than shorter fibers. And what the authors of this paper tried to do is to test this hypothesis and try to prove it empirically. So what they did was to basically measure a lot of axons and myelin. Um, they measured from three different regions of the brain. So they collected data from the corpus callosum, assuming that you would find mainly long fibers there. And then they collected data from the so like the white matter just below the gray matter near the motor and S1 cortex, and between V1 and V2, assuming that these fibers will be short, or at least predominantly. And what they did in the end with an automated strategy was to basically measure up to 400,000 fibers or something like that. So here you can see in the figure one the various sites from which they recorded their data. What they basically had to do is to collect a lot of pictures, microscopic pictures of sections of axons. On top, you can see on the top right, you can see a section of the corpus callosum, and on the right bottom panel, you can see a section of what they generally call superficial white matter, which is a term to group the data collected from M1, S1, and V1, V2. What they define as superficial is less than 3 millimeters from the gray matter. So given this premise, what they found is basically, so what they did and what they found is that basically, yes, short-range fibers tend to have smaller axon diameters and thinner myelin as compared to long-range fibers. And the way they reached this conclusion, let's try to fit this image. Okay, so what they did, they collect the raw data here on the top two rows, left panels. They trained a deep learning model, a dense net, to automatically segment the images and get the borders of the axons and basically the myelin sheet. Here on the right, you see the same picture, just with every axon colored. It's its own color. They then fitted an ellipse into the axon and the myelin the structure, and they got it basically the diameter of these structures. And then they went on fitting a model on these distributions. And what they found is, as I mentioned, yes, apparently, okay, let's focus on the first two panels, A and B. What they found is that for both the axon and the myelin sheet, apparently, yes, the corpus callosum tends to have bigger axons, more myelinated. So here you can see on the left, you have on the x-axis the axon diameter and the number of fiber on the y-axis. In blue, you have the corpus callosum distribution. In yellow, you have the superficial white matter distribution. This on the left is the distribution for the axon diameter. And on the right, you have the same, but for the outer fiber diameter, which is basically the axon plus the myelin sheet. And what you can see is that, yeah, the blue distributions tend to be shifted toward the toward the right in both cases, and they say this is significant. So yeah, long fibers, more myelinated and tend to be bigger. What they also found, which is interesting and also kind of makes sense, is that the G ratio of these fibers is pretty much the same regardless of the group. And the G ratio is simply the ratio between the inner diameter, so the axon diameter, and the overall diameter, so the axon plus the myelin. And these tend to be constant, which, yeah, it makes sense. So smaller axons have like a thinner myelin sheet, and bigger axons have a thicker myelin sheet, but the ratio is pretty much constant. What it changes is that, of course, um, a bigger axon and more myelinated axons tend to have, at least according to the model they used, faster speed of conduction. So they can conduct neural signal faster, which, yeah, makes sense. What they also looked at is the distribution of like a more nuanced distribution of the G ratio in both the superficial white matter here on the left, and in the corpus callosum here on the right. And what they observed is basically that in areas with predominantly long fibers, so the corpus callosum, the G ratios tend to be more kind of uniformly distributed. Well, not uniformly, but the distribution is more compact around a value of like 0.5, 0.6. While in areas like superficial white matter, there's tend to be more variety. So there's kind of a mix of fibers with like a G ratio that is like the center of the distribution is still around 0.5, 0.6. But you also have like more axons with a considerably higher G ratio, meaning you have relatively bigger axons but with a thinner myelin sheet. So the higher the G ratio, basically, yeah, the myelin sheet is smaller as compared to the axon diameter. What else did they found? So the figure 4 is pretty much the same as what I've shown now. It's just more data, a bit more pulled apart. Also, what they did is to fit this GEV distribution, which is based on 3 parameters. I don't know the details. I didn't go much into that, but it's basically a distribution based on a measure of central distribution, a measure of variance of distribution, and a measure of like a parameter of measuring the skewness of the distribution. So you can think about it like mean, variance, or standard deviation and skewness. And what you observe is that, yeah, it's pretty much the same. This is the same image, just in one picture. So you see the axon diameter in the thick line, and the outer fiber diameter in the dashed line. And on the right, you can see the distribution of the parameters. So the mu parameter, which is sort of like the mean or like the central tendency, the sigma, which is the measure of the spread of the distribution, and the xc parameter, which is a measure of the skewness of the distribution. So you can see there's kind of a difference between for the central tendency and the dispersion. So the corpus callosum, like the long fibers tend to have like higher values in these parameters, but for the skewness tends to be the same, which is I guess it's reflected in the fact that regardless where you are, you tend to have the same G ratio. If you look at the axon diameters and myelin thickness. And yeah, that's pretty much the main point of the paper. So short range fibers have smaller axons diameters and thinner myelin as compared to long range fibers. The superficial white matter fibers tend to show more diversity as they have like a bigger span of G ratios. Even though the G ratio is pretty much the same across areas. And yeah, you have different conduction velocity if you look at long fibers or short fibers. Of course, there's many questions still to be answered. Say, what is the relationship with function? There's some conflicting. They also mentioned some conflicting evidences concerning subsections of the corpus callosum, where like the diameter of the fibers there might actually vary across sections. They didn't look at it. They just pulled all the data from the corpus callosum together. And of course, the big question also is like how in development the axonal caliber and myelination are regulated. That's still unanswered. So yeah, that's pretty much the main. These are pretty much the main messages of the paper. So I try to keep it short in case you have questions. So yeah, the floor is yours.
Amazing. Thank you. Any questions, just raise your hands. If you're still pondering your questions, let me get started. Great paper. Thanks for presenting it. I was wondering, they set out with the premise to look at the belief that we have about myelination and axon diameter. But I was wondering what the implications now are of their findings. Like where do you go from here other than widening the mapping to developmental trajectories, for example? Like what's the implication?
Yeah, good question. This is one of the things they left pending. Um, so they point at some interesting possible subdivision, for example, of wide matter distribution between S1 and M1, um, where, for example, in the precentral gyrus there and the postcentral gyrus, there might be subdivisions of relatively thicker and a smaller caliber axon. So like it's possible that fibers going from the precentral gyrus back to the postcentral gyrus are relatively smaller in diameter as compared to fibers that are projecting then from M1 down to basically the spinal cord. So that would have a functional relevance, and it's like, of course, if you have to project all the way down to the spinal cord, you definitely need like bigger axons because you need to be quicker. So yeah, other functional uh, yeah, the functional that I'm just speculating like at this point because they don't go much in detail about this. It's mostly a paper about the microstructure of the brain. Uh, but yeah, one could hypothesize that simply like if you need to cover a longer distance, then you definitely need to be quicker. If you want to keep the time, like if you want to be equally fast in like conducting messages from one point to another, then if you have to cover a longer distance, you have to be faster to maintain the same like difference in time. I don't
So
know if that.
the difference between the short and the long range fibers, do they speculate in any way why the shorter fibers are thinner and less myelinated compared to the longer connections and relate that to adaptability in the brain or plasticity, for example?
No, I don't know. There's nothing really like about adaptability or plasticity.
Okay, thank you. Fernando has a question for you.
Thanks, Adel, for the presentation. I had a question about where exactly did they sample from in the tissues? Did they sample from 4 different places in superficially or from two?
Yeah,
If I
so
know.
they had a few brains, like 4 or 5. And they sample from from M1 and S, the pre-central and post-central gyri superficial white matter, and the areas between V1 and V2 also superficial white matter, and they pulled all these like data together. Same for the corpus callosum, they collected from different sections of it, and they from different brains, and as far as understood, that they pulled all the data together. So yeah, I don't know about like, of course, like there's a consideration about like getting the brain. You need to get them quick and fixate them. Otherwise, yeah, I mean, you can already see that these brains were prepared relatively quick, but you already see like structural like damage. So yeah, different brains, different parts. They pull together everything from the corpus callosum, and they pull together M1, S1, and V1, V2 into the superficial white matter.
Mm-hmm. Uh, and now I have two more questions. Like, why would they make this choice for the superficial samples in particular?
Yeah, this is just an assumption. So the assumption is that if you pick white matter from the corpus callosum, you're sampling basically only long range fibers, because the corpus callosum, yeah, it can be assumed to be containing mostly long range fibers, and the superficial white matter, since we know the U-shaped shorter like fibers tend to connect at the adjacent gyri. So if you sample from the superficial white matter, their assumption was, okay, we're gonna pick mostly short range connections. And since the idea of the paper was to test, okay, are long range fibers different from short range fibers? Okay, let's do this. And we assume that we are sampling short range. Short range fibers are going to be mostly superficial.
Mm-hmm. And this is more like a speculative question, but since there was a distribution in both samplings, that there was a thicker in general uh, curve, well, distribution for corpus callosum, but it was also towards the thinner parts in the more superficial sampled areas. But still, there was in that distribution, I think it's figure 2 or well, figure 3.
Mm-hmm.
Uh, there are still some axonal diameters that are thicker than in the superficial area than in the corpus callosum. I'm wondering,
Yeah, of course.
do you think that the thicker axons may be axons that project further, or do you think that it's just a matter of a distribution that is
Yeah, I mean, of
naturally
course, these are
occurring?
distributions, so of course there is some overlap. And the point is whether like there is a general tendency to be different or not. I mean, yeah, you could speculate. So they do show that like the superficial white matter has a bigger variety in terms of, axon diameters and myelin thickness and so on. And of course, you can also assume that, no matter where you are in the brain, like almost kind of anything that is, even primary cortices, they might get like long range connection. If you think about B1, it definitely gets like long range connections from the optic radiations. Uh, so yeah, I mean, there is some overlap, and the superficial white matter might contain, of course, like long range fibers. It's a bit, yeah, you can speculate about it. It's hard to prove based on these data. You should do like tracing studies and so on. But yeah, the main point is that there's a general tendency for long range connections to be, on average, like yeah, thicker and more myelinated than the short range connections.
Thanks a lot.
Right. I don't know how to pronounce your first name.
Then that's probably me.
Yes.
Sure. It's a Dutch name. Don't worry about it. People butcher it in all sorts of ways.
What's the correct
perfectly fine.
pronunciation?
Sjoerd.
Sure. Okay,
Yeah,
great.
exactly.
Thank you. Welcome. What's your question?
Thanks. Thanks, Eduardo. Very concise presentation. I loved it. Um, my question mostly deals with the variability between the brain donors, because I was reading that there are 5 different brain donors that are also all a bit older, well, relatively young, I suppose, for brain donors. But do they treat, because this seems a lot like a lot of clustered data, do they look at intra individual differences in these comparisons in the white matter tracts in these estimations, or do they address it in their statistics?
Okay, as far as I know, and I didn't go in the details of the method sections, they don't. They pool all the data together. And what they have is, yeah, 5 brains for male uh, age, yeah, all above 60, so 61, 74, and so on, with relatively short postmortem delays.
Yeah.
But yeah, no, as far as I know, maybe in the method sections they mentioned something, but I'm not sure. So as far as the main section of the paper is concerned, that's just pulling together all the data. Also because they do insist, like there are a few papers doing something similar to these on animal models. But apparently this is the first paper doing something this vast, like measuring up to 400,000 fibers, like using an automated approach. And the previous studies were they used more like a manual kind of dissection and like fiber definitions. So yeah, I don't think they go that down into variability
Well,
between samples. Also because it's
well,
really a very few brains.
well, I understand they have constraints, but it would have been very nice, of course, to see this same kind of distribution or this pattern with each of the different individuals rather than just.
Yeah, no, agreed. Yeah, that will be
I
definitely
mean, I at
interesting.
least treat it as such, right? Or at least treat the samples from different subjects as
Yeah.
a variable in some of the analysis. I think that would have made it maybe a bit more powerful.
Yeah. I mean, they do make the point in the discussion that, for example, yeah, as I mentioned, like different sections of so what they did is to assume that the corpus callosum is pretty much homogeneous, even though there are like, yeah, it's still debated. There are like, people disagree on whether there's a gradient or there are different pattern of distributions about like concerning the axon diameter and the myelin in the corpus callosum. So what they did, they do say, we're just gonna consider for the sake of this paper, we just want to set the ground and we're just gonna pull all the data of the corpus callosum together, and then future studies might look into that. So maybe they are looking into this. We don't know.
Yeah.
Hopefully. But yeah, it would be interesting. On the other side, I don't know, with 5 brains,
Well, you can
yeah.
also conclude that it doesn't. Yeah, that you
Yeah, I
don't.
mean they do have a lot of data, so maybe from like for 400,000 like divided by 5 is it's like 80, yeah, it's like nearly 80,000 fibers per brain on average. So yeah, maybe you have like fewer samples, but if you have this massive approach, like collecting a lot of data from every sample, actually maybe variability is not that bad of an idea. So yeah, let's hope for a new publication.
Thanks.
Variability is never a bad idea to look at.
No, it's never bad, but the way you do it, it's relevant.
But did I understand you right that you just said the corpus callosum is homogeneous in
Well,
its?
they assume it to be, even though there are evidence that might not be the case in terms of axon diameter and myelin thickness.
There's quite accumulation of evidence that you have a variability in the diameter across the corpus callosum, right? So
Yeah,
that
I'm
is
not an expert on that, so I'm just taking for.
okay. I don't want to take time away from Lilith, who also has a question.
Yeah, hi Eduardo. Sorry I didn't come from the beginning. There was a misunderstanding in schedule, but like I have a question. I'm not sure if you told about this before. So as far as I know, there are a lot of axons that are not myelinated. Do they discuss about it in this paper? Maybe there are some in those regions that they sampled. Because in my mind, I have like a number like 70% that they're not myelinated or something like this, and we cannot capture them properly with a tractography. So I was wondering, do they talk about that?
Okay, I'm not sure about the figures here, but I know there are axons that are they're mostly non-myelinated. I'm not sure about the central nervous system, though. I guess most of them they should have at least like a thin layer of myelin. But yeah, I don't know. Oh yeah, tractography. I mean, tractography is mostly picking up anisotropy defined by the axon, but yeah, so this is completely different. This is like microscopic data, and what they're looking at, if you look at the pictures, it doesn't really matter whether an axon is myelinated or not, or how much. What the model is segmenting is the basically the inner the diameter, which is assumed to be the axon. So the bluish, the light blue, green, whatever color you want to call it. So that's assumed to be the axon, and then the yellow border, no matter how thick it is, that's supposed to be the myelin sheet.
Yeah, but for example, I have never seen axons in microscopes. I don't know if I'm seeing things right. But for example, in the figure A, left lower part, there is like sort of a circle. I don't know if this is a I don't know how the model works. So does it maybe captures those only that has myelin and then but yeah, I guess it's something you need more discuss go deeper in the way they do it. But it seems like there is a circle, figure A, if you go upper with your upper, figure A,
Yeah, sure. Yeah.
lower left. Left,
Like
left.
this
Yeah,
structure here.
what is it, for
Yeah,
example?
I don't know. I'm not a microbiologist, but
Yeah, me
yeah.
too,
So
but
the first of
in a,
all, the deep learning approach, this like they used an automated segmentation approach, which is basically a deep learning model. I also didn't go into the details. I assume it's a convolutional neural network for image segmentation. Yeah, I mean, what you is like a more interesting example is also like how good this model is. Probably it has a pretty good accuracy, but yeah, you still get like if you
no,
look at
because
this.
see, for example, also if you go a bit left to with your hand,
Uh,
left.
Sorry, yeah.
Yeah, see, there is an empty
Yeah, this
spot,
structure here.
and there's
Yeah, I
also
don't
like
know.
some blobs. I don't know if it's just the model not capturing the unmyelinated ones, but
Yeah, I don't even know whether this is an accent or not. But
me
I
too. So,
guess,
but that's why I said I've never seen a
yeah, I don't
micro.
know because I'm not a microbiologist or again, microanatomist or. But I mean,
Yeah.
you can also see like here in this section, you have maybe two struct, maybe one. This is, but and this is segmented as kind of one bigger. So definitely, these kind of models, they have pretty high accuracy for sure because most of the accents are nicely segmented. You're
No,
never gonna
yeah,
be 100% sure.
yeah.
So what you leverage, but the alternative on the like at the other hand of the distribution, you have manual labeling and just drawing
No,
like
no, no.
manually stuff. So
That's going to be insane.
what I want to assume here is that if you have these amount of data, like 400,000 like data points, then if you have a few of them that are mislabeled, that doesn't really matter that much.
No, 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 yes, but for sure, this method seems doing great with myelinated ones. I was wondering if
I
it is
mean, it
possible to
does
train.
pretty.
I was wondering if it's possible to train that type of model to detect some sort of unmyelinated bonds. But
I mean, yeah, probably. I mean, it's a convolution. I'm
yeah,
assuming it's a convolution
at the
neural
moment
network,
they can do
so
everything.
depends on the quality of your image, depends on your data. But I mean, it does pretty
Yeah.
okay even for like small, like kind of small, probably not very myelinated axons. Just looking at these pictures that probably were picked because they are pretty good. I don't know. But yeah, like this one is segment, like these small axons with not much myelin are, yeah,
Yeah.
kind of segmented. try to match them on the image. Seems pretty okay.
No, yeah.
And then of course, I mean, also like in the first place, you have also to consider that these images are pretty good, but they already suffered like structural damage from the fact that the brains were fixated and then cut into pieces and what that, yeah.
Yeah.
So it's a messy
Okay.
procedure.
Thank you, Eduardo.
Yeah. Lovely discussion.
I'm still stuck on the homogeneity, and for those of you who can see the chat, can see that Evie and I were.
Yeah, you're free
Okay,
to go there and look. They mentioned these. They have references.
Yeah. So there
Um,
are a bunch of papers looking at that. So it's strange they say that it's homogeneous because
well, they don't say it is. They just say, well, for the sake of this paper, we assume it is because
it's better.
Right, but if you change your underlying anatomical assumption, what do you then do with the results? That's the thing where I get stuck in my thinking.
Yeah. Well, so the picture you
We
have
know
is
we know the corpus callosum is heterogeneous, not homogeneous.
yeah. Do we have data from diffusion, right? From this, or is it also
Oh, no,
microscopic?
that's histology. That's
Histology, okay.
actual measurements.
But maybe they just took a little piece and within that piece it is homogeneous. Or what? Or do they say that this is how the whole corpus callosum should be done?
So in the chat you see the classical work by Aboid.
Yeah.
We also shared a nice paper from the group with Innocenti. And you can see that there is, for lack of a better word, a gradient of.
No, yeah, I was wondering if within this paper, the one that Eduardo is presenting, they took one little piece and within that piece it is homogeneous, which would make sense.
Right, so one section you mean?
Yeah. But then it will be wrong then, I think, based on those papers that you shared in the chat, that it's not probably the same along
So
the
I
way.
guess you can't even generalize within one brain, right? Along the entire structure of the corpus callosum.
Yeah.
So that's a little bit puzzling to me. But we don't have enough time to disentangle the whole story here. So thanks for joining us today. Everyone in Bordeaux, I see you later. Everyone in Nijmegen, I see you again on Wednesday. Thanks for joining. Thanks for presenting, Eduardo, and have an exciting week in science. Bye.
Thank you.
Ciao.
Bye-bye.